Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 1 Multiple Choice, October-November 2021, Paper 12. Question number 1. A scale bar on an electron micrograph is 2 centimeter long and represents an actual length of 1 micrometer. What is the magnification of the electron micrograph? We know magnification is equals to image divided by actual. So the actual is 1 micrometer. So the magnification for 2 centimeter, it will be 20 millimeter multiplied by 1,000. So 20,000 divided by 1. So the answer will be 20,000. 1C is the correct answer. Question number 2. The eyepiece of a microscope is fitted with an eyepiece graticule and a stage micrometer is placed on the microscope. Which statement about the stage micrometer scale are correct? The scale can be used to measure the actual length of cells directly. No, we cannot use it directly. All right. So A and C is wrong. The scale allows you to calibrate the eyepiece graticule. Yes. Less of the scale is visible as the objective lens changes from 10x to 40x. Yes, this is also correct. So option 2 and 3 correct. Question number 2, B. Question number 3. 4 students were asked to match the function and the appearance of some cell structure in an animal cell. The functions were listed by number. mRNA passes through the ribosome, organizes microtubules to produce the spindle during cell division, synthesis of polypeptides. All right. So mRNA passes through the ribosome. All right. That's basically passes through to the ribosome. So this is nuclear pores. All right. Organizes microtubule to produce the spindle during cell division. This is centriole. This is centriole. Synthesis of polypeptides. This is ribosome. The appearance were listed by the letter. Which structure correctly matched the numbered function with the appearance of the cell structure? So for point number 1, we have V and X. mRNA passes through the ribosome nuclear pore. So let's look at V, membrane which surrounds an enclosed inner cavity. And X, a double membrane interspersed with pores. So X will be the correct answer. For question number 2, W or Y? So W, non-membrane bound spherical structures. All right. And non-membrane bound cylindrical structures. Well, centrioles form microtubules, which are cylindrical structures. So Y is the correct answer. So last option for number 3, synthesis of polypeptide, W, non-membrane bound spherical structure, which is ribosome. All right. Question number 3, answer D. D is the correct answer. Question number 4. Which cell structures are required for the formation of lysosomes and hydrolytic enzymes that they contain? All right. Of course, formation of lysosome requires energy, so mitochondria will be needed. Rough endoplasmic reticulum will be needed for the protein synthesis. Golgi body will be needed for the packaging, so B will be the correct answer, 4B. Question number 5. Which cell structures contain ribosomal RNA? Ribosomal RNA will be contained by chloroplast and mitochondria because they are early bacteria. Ribosomes are synthesized within nucleus. So ribosomal RNA will be found in nucleus. Smooth endoplasmic reticulum does not contain ribosome. So 1, 2, and 3. 5B will be the correct answer. Question number 6. The single-celled organism Theo Margarita namibiensis measures 700 micrometer in diameter and is enclosed in cell wall. All right. The genetic material T. namibiensis is located free in cytoplasm, where it occurs as thousands of copies of circular DNA. So that means circular DNA, it indicates of that of a bacteria or prokaryote. Most of the cell is taken up by large vacuole, which stores essential chemicals for metabolism. Which statement is correct? It must be a eukaryote? No, it cannot be a eukaryote. It must be a plant. No, it cannot be a plant because it has circular DNA. It must be a plant because it is enclosed in cell wall. No, even though it is enclosed in cell wall, all right, it has circular DNA. It must be a prokaryote because its DNA is circular and located in the cytoplasm. Yes. 6D. D should be the correct answer. All right. 7. Question number 7. A student carried out 4 tests on a sample of biological molecule. Emulsion test went cloudy, meaning fat present. Benedict's went yellow, meaning reducing sugar present. Violet purple, meaning protein present. Iodine yellow, no starch. Which conclusions made by the students are correct? Is fat present? Yes. Glucose was present? Cannot tell. Protein was present? Yes. Starch was not present? Yes. All right. So 1, 3, and 4. The student can conclude on comments of 1, 3, and 4. 7C is the correct answer. Question number 8. Which general formula is correct for monosaccharides such as fructose? Fructose has a C6H12O6 as a general formula. So, uh, obviously, if we shorten it out, it will be CH2O. So CH2O, B will be the correct answer. 8B is the correct answer. Question number 9. Which statements can be used to describe the structures of cellulose? A polymer of glucose monomer linked by 1,4. Okay. Uh, yes. Cellulose is a polymer which is linked by 1,4 glycosidic beta bonds. That is correct. A polysaccharide of hexose monomers. Yes, it is a polysaccharide of hexose monomer. An unbranched macromolecule made of beta glucose monomer. All points 1, 2, 3 are correct. 9A is the correct answer. Question number 10. Complete hydrolysis of polysaccharide requires all the glycosidic bond between the monomers to be broken. Enzyme X only breaks alpha 1,4 glucose. Which row shows how completely enzyme X can be hydrolyzed? Enzyme X can hydrolyze molecules of glycogen and amylose. All right. Negative indicates no hydrolysis. Only 1 plus means some hydrolysis and 2 plus means most hydrolysis. Since this particular enzyme can break down only 1,4 glycosidic bond, so for amylose, there will be full hydrolysis. However, for glycogen, there will be a partial hydrolysis. So 10A is the correct answer. Question number 11. Which molecules contain at least 2 double bonds? At least 2 double bonds. A saturated fatty acid has 1 carbon-carbon double bond because, as you can see, there is only 1 carbon-oxygen double bond and the remainder are saturated. So no double bonds. Saturated fatty acids, saturated fatty acids. Hemoglobin and collagen molecules has more than a CO double bond, more than 1 or more than 2. All right. So because every protein molecule in every amino acid, there is a CONH bond. Question number 12. The diagram shows an amino acid. Which group is changed to produce different amino acids? All right. So we know that the CH3 group will be changed because that is the R group. This is the R group that is represented. So CH3. Question number 12. C is the correct answer. Question number 13. Which row is correct about protein structure? All right. So if we look, when answering question like this one, it's important that we choose for this quaternary first because most amount of mistakes will appear in this particular section. Always formed by association of 2 or more polypeptides. It can be similar, it can be different, correct. Always formed by 2 or more polypeptides held together by bonds between the polypeptides. Well, no, it's not necessary that they must be held together by bonds between them. Always formed by 2 or more different polypeptides to give the final shape. It is not necessary to have different polypeptides. Same polypeptide repeated unit can form a quaternary structure as well. All right, always formed by two or more polypeptides and a metal ion. It's not necessary all the time metal ion will be incorporated in the in the protein structure, in the quaternary structure. So option A is the correct answer. All right, the primary structure is determined by the sequence of DNA nucleotides, correct. Occurs by hydrogen bonding between CO and NH group of amino acid in the secondary structure, also correct. Tertiary, disulfide ionic hydrogen bonds and hydrophobic interactions are all involved. Yes, this is correct. So question number 13A is the correct answer. Question number 14. Which graph correctly shows the possible changes in energy as a chemical reaction progresses with or without the enzyme? Okay, so we can see substrate, we can see product. All right, with or without the enzyme, they must end in the same level. They must end in the same level. All right, then we can see chemical reaction with without the enzyme, energy levels as chemical reaction progress. All right, so substrate, progress of reaction. All right, we know that without enzyme, without enzyme, the reaction requires a higher activation energy. But with enzyme, the reaction requires a lower activation energy. So C should be the correct answer. Question number 14, C is the correct answer. 15. The graph shows the relationship between the concentration of substrate and the rate of enzyme catalyzed reaction. The rate of enzyme catalyzed reaction, concentration of substrate increasing in this direction. Which row shows Km and Vmax for the enzyme would be affected if the same reaction was carried out in the presence of a competitive inhibitor? We know that in presence of a competitive inhibitor, there is a competition. And as a result, the Km value is actually increased. However, the Vmax remains the same. So B should be the correct answer. Question number 16. Liver cells contains vesicles that have proteins in their membranes, which are specific for the transport of glucose. When these cells need to take up glucose, the vesicles fuse with the cell surface membrane. How does the uptake of glucose occur? So the uptake of glucose, we know that uptake of glucose occurs by means of facilitated diffusion in liver cells. So 16, the answer should be D. 17, a student set up a experiment to investigate diffusion. A agar block 1 cm by 1 cm by 1 cm was stained uniformly with a water-soluble blue dye. The block of agar was put into a test tube containing 10 cm cube distilled water at 20 degrees Celsius. The intensity of the blue color of the water after 5 minutes was measured. Four other experiments A, B, C, T were then carried out using different numbers of agar blocks, different sizes of agar blocks, and different temperatures. All other variables were standardized. Which experiment would give a lighter blue color in the water after 5 minutes compared to the first experiment? Well, if we compare to the first experiment, we must all, of course, eliminate the higher temperature. All right, because at higher temperature, there will be more amount of pigment that will come out. All right, so after 5 minutes, all right, now we have 0.5 into 0.5 into 0.5 block, but there is only two blocks, and 2 into 1 into 1, and there is one block. Definitely there is greater surface area which is exposed in experiment D. All right, but there is lower surface area which is lower surface area which is exposed in 1 in B, because the number of agar blocks is 2. All right, so the answer should be B. uh all right, all right, because there is less amount of agar blocks that has the color. So definitely B will be the correct answer. 17, B is the correct answer. 18, which description is correct? A centromere holds two chromatids together until the end of prophase and attaches to the microtubules of the spindle. Well, at metaphase, all right, centromeres, you know, they attach themselves with the microtubules of the spindle. So A is wrong. A chromatid is one of the two identical parts of a chromosome and is made of proteins and two DNA molecules. All right, chromatid. All right, okay. Question number 18. Well, they're not made of proteins, so they should be, then next, a chromosome is a structure with two identical parts made of DNA and proteins found in the nucleus of the prokaryotic cell. All right, a chromosome is a structure with two identical parts made of DNA and protein found in the nucleus of prokaryotic cell. So we don't find chromosomes in prokaryotic cell. This is wrong. Next, a telomere is a sequence of DNA nucleotides such as G, 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 T, A, repeated many times and found at the end of each chromatid. This is a correct statement. All right, so 18, D is correct. Question number 19. During metaphase, a scientist stains the chromosomes of a diploid animal cell with fluorescent dye to allow the telomeres to be observed. This cell has 26 chromosomes. How many telomeres will the scientist observe? So if the cell has 26 chromosomes, each chromosome will have four telomeres. So 26 into 4, 26, 104. D will be the correct answer. Question number 20. The photomicrograph shows cells in various stages of cell cycle. Which cell contain twice as many DNA molecules as the cell from the same organism that has just finished complete mitotic cell cycle ending with cytokinesis? All right. So this is a cell at metaphase. This is a cell at anaphase, prophase, anaphase. So in all of these cells, there will be double the number of DNA molecules. 20A will be the correct answer. Question number 21. Uncontrolled cell division can result in the formation of tumor. What part of the cell cycle would take less time during the formation of tumor? In tumor formation, interphase is the phase that is the shortest amount of time. So 21B. Uh, Question number 22. A piece of DNA molecule contains 84 bases. The table shows the number of adenine and cytosine bases in one or both of the DNA strands in this piece of DNA molecule. Adenine, we can see adenine, we have 28 in strand 1. So there is a very likelihood that if there is adenine, which is 28, all right, there will be thymine in strand 2, which will be 28. All right. If there is, a 23 adenine in strand 2, there will be 23 thymine in strand 1. Cytosine and guanine. In terms of guanine, there is 15 cytosine in strand 1. So there will be 15 guanine in strand 2. All right. Now, the total molecule should be 84 base pairs long. So 23 plus 28 plus 15, and then 84 minus all of this equals to, the answer is 18. So there will be 18 cytosine on the strand 2, and there will be 18 guanine in the strand 1. How many guanine bases are present in these pieces of DNA molecule? So guanine, there will be 18 plus 15, total of 33. So B will be the correct answer. Question number 22, B is the correct answer. Question number 23, the mRNA sequence of 3 stop codons are shown. Which mutation in the template transcribed strand of a DNA sequence that codes for a polypeptide would cause translation to stop prematurely? All right. So the template strand of DNA sequence, ATT, all right, change to ATC. All right. So ATC, ATC will be A will be U, A, G. All right, ATT. ATT was U, A, A. So it was already a stop codon, it is going to a stop codon. So this is not gonna stop prematurely. ACT changed to ACA. ACT will be U, G, A. UGA is already a stop codon, so should be cancelled. ACC. ACC is UGG. UGG is not a stop codon. UGG changed to ATT. All right, ACC changed to ATT will be UAA. All right, C should be the correct answer. If ACC changed to ATT, then stop codon will be there prematurely. Question number 23, answer number C is the correct answer. 23C. Question 24, which features of companion cells are essential to their function? They are connected to the sieve tube elements by plasmodesmata. Okay. They have thicker cell wall than a sieve tube element. All right. Companion cell are essential to their function. Number 2 is not essential to their function. Number 2 gone, meaning A and D wrong. They contain a nucleus and mitochondria. Of course, they contain a nucleus and mitochondria. So for 24, option B should be the correct answer. 24B. 25. Which statements about water movement in plants are correct? Water can pass through the cellulose cell wall. All right. We know that water can pass through cellulose cell wall. Water cannot pass through lignified cell walls. Also correct. Water can pass through cell walls that contain suberin. No. All right, so 1 and 2 is correct. 25B is the correct answer. 26. The diameter of a tree trunk usually decreases slightly during the day. Which changes in environmental factor during the day could cause the diameter to decrease even more? Increase humidity? Well, it will decrease transpiration. Increase wind speed will increase transpiration. Increase temperature will increase transpiration. So B should be the correct answer. Question number 27. Which sequence of events could lead to mass flow in phloem sieve tube? Sucrose moves into phloem sieve tube element. Proton and sucrose molecules move into companion cell through a co-transporter protein. A very high hydrostatic pressure is produced. The water potential of sieve tube element decreases. Okay. Proton H plus ions are actively pumped out of a companion cell. Water moves down a water potential gradient by osmosis. We know from all of these activities, number 5 is the first activity that should be done. H protons are actively pumped out of a companion cell. So if 5 is the first statement, then 5, 2, 1, 4, 6, 3 should be the correct answer. 27D is the correct answer. 28. What will happen as a result of blood pressure in the right ventricle becoming higher than the blood pressure in the right atrium? Okay, so question number 28. What will happen as a result of the blood pressure in the right ventricle becoming higher than the blood pressure in the right atrium? All right, when the right ventricle pressure will be higher, then the atrioventricular valve will close. The seminal valve in the aorta will close. No. The seminal valve in the pulmonary artery will close. Probably not, because it should open at this particular point. The left atrioventricular valve will close. No. The right atrioventricular valve will close. Yes, D is the correct answer. 28, D is the correct answer. Question number 29. The graph shows the pressure changes in different parts of the heart during a mammalian cardiac cycle. W, X, Y, Z indicate when a valve opens or closes. Which row correctly identifies W, X, Y, Z? W. At W, atrioventricular valve closes. So Atrioventricular valve opens, semilunar valve opens. These are going to be cancelled. Atrioventricular valve closes. B is the correct answer. And let's check for the other ones. At X, semilunar valve opens, correct? All right. At Y, semilunar valve closes. At Z, atrioventricular valve opens. So question number 29, B is the correct answer. Question number 30. Which statements are correct? Number one, compared with blood, tissue fluid has less protein and no red blood cells. Compared with blood, tissue fluid has less protein and no red blood cell. This is a correct statement. All right, lymph may contain lipids, carbon dioxide, phagocytes. Yes, lymph may contain them. Tissue fluid contains glucose, amino acid, urea, carbon dioxide. Yes, 1, 2, 3, all correct. 30, option A. Which statements about the formation of hemoglobinic acid are correct? It can only occur when the dissociation of oxygen with the dissociation of oxygen from hemoglobin. Correct. It removes excess hydrogen ions, preventing blood from becoming too acidic. Correct. It is linked to the action of carbonic anhydrase. Also correct. So 31, option A will be the correct answer. Question number 32. What explains how the maximum volume of oxygen is taken up as blood passes through the capillaries of the lungs? Each hemoglobin molecule can temporarily bind to 4 oxygen atoms. Probably, but why would that? Okay. How maximum volume of oxygen is taken up through the capillary of the lungs? Yes, could be correct. The dissociation of carbon dioxide from carboxyhemoglobin allow more hemoglobin to be available for binding with oxygen. All right. Okay. The binding of the first oxygen molecule to hemoglobin decreases. This is a wrong statement. Actually, binding of the first oxygen with hemoglobin increases the affinity. Oxyhemoglobin formation increases the ability of red blood cells to transport oxygen. Yes, so this is the right statement which explains how the maximum volume of oxygen taken up by as the blood passes through the capillaries of the lungs. Because oxyhemoglobin, if hemoglobin binds with one oxygen, then the HbO2 increases in affinity. The HbO2 increases in affinity to bind with more oxygen. And the HbO4 also increases in more affinity to bind with more oxygen. And finally, that leads to fully dissociated hemoglobin. So increases affinity. Increases affinity. All right. So D is the correct answer. Question number 33. The image shown is a photomicrograph of a transverse section of a part of the gas exchange system. Okay. We can see ciliated epithelial cell. We can see ciliated epithelial cell. All right. What describes the image? What describes the image? A thin inner layer of ciliated epithelial cells on top of layer containing cartilage, supported by elastic fiber. So we can see a thin layer of ciliated epithelial cells on top of layer containing cartilage. Well, the cartilage is over here. This is cartilage. So this is wrong. A very thin epithelial lining, a very thin epithelial lining with walls containing elastic fibers, surrounded by many blood vessels. We don't see a lot of blood vessels. An inner layer of ciliated epithelial and goblet cells on top of elastic fibers, supported by an outer layer consisting of cartilage. Yes, we can see ciliated epithelial, we can see goblet cells on top of elastic fiber. This is the elastic fiber. We can see supported by an outer layer consisting of cartilage. We can see the cartilage. C, question number 33, C is the correct answer. Question number 34, which row shows the tissue that are present in the wall of the trachea and the wall of the bronchus? In the wall of the trachea and the bronchus, there will be smooth muscles. Squamous epithelial will be absent. Goblet cells will be present. So C should be the correct answer. Question 35. When a person suffers an asthma attack, the tubes of the gas exchange system narrows and extra mucus is produced. Which changes occur during an asthma attack? Activity of ciliated epithelium increases, definitely. Endocytosis of goblet cell increases. No, exocytosis increases. So point number 2 is wrong. Smooth muscles are more active. Yes, they contract. So 1 and 3 correct. Question number 35, C. 36. Which flow diagram correctly describes the effect of tar entering lungs? Carcinogens come into contact with DNA. Mutation occurs, uncontrolled cell division. Could be. All right. Goblet cells secrete more mucus. Mucus accumulates, causing infection. Phagocytes attracted by inflammation. All right. Mucus accumulates, causing infection. Phagocytes attracted by inflammation. Elastase destroys the alveolar wall. Okay. Mutation occurs. Phagocytes attracted by inflammation causes irritation and coughing. All right. So after mutation, it talks about irritation and coughing, so should be canceled out. All right. Goblet cells secrete more mucus. This is correct. However, mucus accumulates, causing infection. Phagocytes attracted by inflammation. It's not only the effect of tar entering the lungs. The effect of tar entering the lungs should be because it's a carcinogen, it will come in contact with DNA. As a result, mutation will occur. As a result, uncontrolled cell division will occur. So 36A is the logical answer. Question 37. Which control measures would reduce the transmission of tuberculosis? Tuberculosis, boiling water before drinking it? No, it's not going to stop tuberculosis. Antibiotics, yes, it's going to stop tuberculosis. Vaccination, definitely. So D, 37, D is the correct answer. 38, comorbidity is where an individual has two or more diseases or medical conditions at the same time. Some medical conditions are particularly likely to result in comorbidity. In one example, initial infection with one pathogen can increase the risk of developing a disease by a factor of 15 or more. The second disease is then the major cause of death for these people. Which description could fit this example? Oh, cholera infection followed by development of malaria? No. Right. Cholera infection followed by development of AIDS? No. Cholera does not cause malaria. Cholera neither, it can cause AIDS. HIV infection followed by the development of TB? Yes. This can be leading cause of death. 38C. Which conditions, 39, which conditions are infectious and result in high white blood cell count? All right, so leukemia is not infectious. All right, okay. And myasthenia gravis, it is not infectious as well. So Only TB is the correct answer. D is the correct answer. Which cell type? Which cell type causes, question number 40, which cell type causes the secondary immune response to be much faster than the primary immune response? All right. So memory cells definitely allows secondary immune response to be much faster. So 40B is the correct answer. Guys, we will be solving videos like this and posting it in our channel every day. If you have any questions that you want to solve, all right, okay, please let us know in the comment and subscribe to our channel, and you will be notified. Thank you very much for joining the class today.